In this video, we're going to look at protein synthesis. So protein synthesis occurs in the nucleus and the cytoplasm. It creates peptide chains from the DNA template. And it is divided into two steps, one of them transcription and the other one translation. So overall, the DNA molecule itself is then converted into RNA, or a copy is produced in RNA, and then that codes for amino acids. So the first step is that we have transcription. Transcription occurs in the nucleus of the cell where the DNA is found. Now, RNA polymerase binds to the promoter region of DNA. This is usually before a single gene of a chromosome. The second step is that we have elongation. This is where free mRNA nucleotides base pair with complementary bases on only one of the DNA strands, which is the template strand. The third step in transcription is we have termination. Now, termination is where we have the new strand of mRNA that detaches from the DNA template and leaves the nucleus via a nuclear pore. Okay, these three steps are really important for us to have that mRNA, the messenger RNA, in order for us to produce the amino acids in the next step of translation. So translation occurs in the cytoplasm of the cell once that mRNA has left the nuclear pore and now has entered the cytoplasm. This is where we have free transfer RNA molecules that have the unpaired bases, which are the anticodon that attach to the codon or the triplet of the mRNA molecule. So tRNA also has an amino acid binding site to it, which is right at the top of the clover shape tRNA molecule. The mRNA molecule binds to a ribosome via its small subunit. Okay, because the ribosome has a small and a large subunit, the mRNA itself binds to the smaller subunit. The first three exposed bases, or codon, are usually AUG, and this is the start codon. So the tRNA molecule will complementary anticodon or base pair with the AUG so that UAC, which is the complementary base, will therefore code for the amino acid, which is methionine, and that one will start off the overall translation. So here is an image just to represent what we just talked about before. We have the small ribosome subunit and then the large ribosomal subunit here. We have the mRNA molecule here with the anticodon from the tRNA. Now the anticodon here is then base pairing to the mRNA molecule so that we have UAC as the start code, which is coding for the MET or methionine amino acid, which is going to start off the translation of the protein overall. Okay, we could just see that the enzyme peptidyltransferase will form a peptide bond between the amino acids, okay, between these ones here. So we have the correct base pairing of the tRNA and the mRNA. We have the amino acids being produced. We have the peptide bonds formed between the amino acids and the overall protein produced. Okay, so we have something called the genetic code, and when we talked about the triplet code before, okay, this is what we're talking about. So those triplets code for different amino acids. Say, for example, we looked at methionine as being the start amino acid before. Okay, methionine would be right here, AUG. Now notice that AUG is the only triplet that codes for the start amino acid methionine. Okay, all the other ones, we have a different amount that can code for that amino acid. Like in this case here, we have 4 different sequences that can code for this one, so this one, so can this one, so can this one, etc. We have a few different stop triplet, okay, which can code for the stop codon. We have UAA, UAG, which is the stop, or we could have UGA for our stop here as well. Now, the way we read these charts is that we start with the left-hand side here. So we go A, for example, is our first codon. Then we look at the U, which is the second one up the top. Okay, and then we look at the right-hand side. So, for example, we're looking at AUG, we're looking for A and then U, and then we're going down to G, which is here, and this one is then AUG. Okay, so in summary, we first of all have DNA replication. Now, the DNA replication itself can produce new DNA, which is actually used for our original template. Now, the DNA molecule itself is that template, and we need to make a copy of that one, so we have mRNA. mRNA is made with the first step, which is transcription or RNA synthesis. This occurs inside the nucleus. The mRNA molecule that is now exits the nucleus and then goes into the cytoplasm of the cell so that translation can take place, or protein synthesis. We have the small and the large subunit of the ribosome working together so that the mRNA will code for those amino acids. The amino acids join together so that we have a protein being produced. So DNA goes to RNA, RNA goes to protein. And that does it for the video.